Hello everyone, welcome to Learn Classic Education, and today we are going to discuss about the TCA cycle, which is otherwise known as the Kreb cycle or citric acid cycle. Okay, so today we will explore this topic, that is citric acid cycle, right? So this cycle is a crucial part of cellular respiration, which is mainly occurs in the mitochondrial matrix, right? So the cycle's primary function is to generate high energy molecules named as NADH, FADH and GTP or ATP. Basically the GTP is converted into the ATP, which is essential for ATP production in electron transport chain or ETC, right? So now let's dive into the topic, right? So this all step of TCA cycle occurs in the mitochondrial matrix, right? So the first step includes the formation of citrate. Citrate is a 6 carbon molecule, okay? And then uh, s- acetyl coenzyme A, which is a 2 carbon molecule, plus oxaloacetate. These two combines to form citrate, which is a 6 carbon molecule, right? So the key point that to remember in this step, because it is a very important step and it is an irreversible step and a regulatory key regulatory point, right? And the enzyme which is involved is citrate synthase. Now the second step is the isomerization of citrate to isocitrate, right? And the enzyme involved in this is aconitase, right? And the citrate is rearranged into isocitrate through a cis aconitate intermediate, right? So this step is basically important that it prepares the molecule for the first oxidation. Then we have oxidation and decarboxylation, right? The enzyme which is involved is isocitrate dehydrogenase, and the reaction is like isocitrate undergoes oxidation and decarboxylation, which forms alpha ketoglutarate, which is a 5 carbon molecule, okay? And the CO2 is released. That is the thing to remember is that it is the like first carbon is lost in this cycle, right? And it is the rate limiting and the regulatory step, very important point to note down. Then we have the fourth step, okay, that is the oxidative decarboxylation. Enzyme involved in this is alpha ketoglutarate dehydrogenase complex, right? And the reaction is something alpha ketoglutarate undergoes oxidation and decarboxylation that forms succinyl coenzyme A, which is a 4 carbon molecule, okay? And the second carbon is released. So the first carbon is released in the third step, and the second carbon is released in the fourth step, okay? The key point is that this is the irreversible step, okay? Now moving to the fifth step, that is the conversion of succinyl coenzyme A to succinate. Succinate is a 4 carbon molecule, right? And the succinyl enzyme A synthetase is the enzyme that is involved in this step. Reaction is something like succinyl coenzyme A is converted into succinate, releasing GTP, which is further converted into the ATP. And this is only the substrate level phosphorylation in the TCA cycle. Very important point to remember, okay? Oxidation of succinate to fumarate, which is a 4 carbon molecule, is the sixth step, okay? And the enzyme involved is succinate dehydrogenase, and the reaction is succinate is like oxidized to fumarate, which is reducing FAD to FADH2, right? And one FADH2 release how many ATP? Please comment down below. And the key point is like this enzyme is also a part of electron transport chain, that is complex 2, that we'll discuss in the further parts of etc, okay? Then the seventh step is the hydration of fumarate to malate. Malate is a 4 carbon molecule, okay? And the enzyme involved in this is fumarase, and the fumarase undergo hydration to form malate, right? And after that we have the last step, which is the final oxidation step of malate into oxaloacetate, and oxaloacetate also is a 4 carbon molecule, and the enzyme involved in this is malate dehydrogenase, and the malate is oxidized to regenerate oxaloacetate that produce NADH. and now the cycle is ready to begin again. And this was all about the citric acid cycle. Now, what about the energy yield per TCA cycle? Could you let me know? Okay, pause the video and think some things about it, right? So, we get three NADH molecules, right, via ETC. So one NADH molecules gives 3 ATP. So we have 3 NADH molecules which is going to give us 9 ATP. Then we have one FADH2 that produces 2 ATP, right, via ETC, and we get one GTP which is further converted into ATP and giving 1 ATP. So total ATP yield per acetyl coenzyme A is 12, and we know that per glucose, since 2 acetyl coenzyme is produced per glucose, so there are 24 ATP involved in the TCA cycle. Okay, and now the regulation of TCA cycle. So the key enzyme is like citrate synthase, that is inhibited by ATP, NADH, and citrate. Then we have isocitrate dehydrogenase, that is activated by ADP, inhibited by ATP and NADH. Then we have alpha ketoglutarate dehydrogenase, that is inhibited by ATP, NADH, and succinyl coenzyme A. Then what about the energy state of the cell? High ATP or NADH inhibits the cycle, and low ATP or NADH activates the cycle. Okay. Then the clinical significance includes the thiamine, which is vitamin B1 deficiency, right? It causes Wenck's Corsac syndrome or the beriberi, that impairs alpha ketoglutarate dehydrogenase, leading to energy depletion, especially in the brain. Okay, then we have fumarate deficiency that causes many metabolic disorder that lead to developmental delays, right? So let us revise the product of citric acid cycle. Let's take a step back and do some mathematics and tracing the fate of the carbon that enter the citric acid cycle and counting the reduced electron carriers, that is NADH and FADH2 and ATP, which are produced. So, in a single turn of cycle, two carbon enters from acetyl coenzyme A and two molecules of carbon dioxide are released. Okay. Second point, the three molecules of NADH and one molecule of FADH2 are generated, right? And one molecule of ATP or GTP is produced. So these figures are for one turns of the cycle, right? And each glucose molecule produce two acetyl coenzyme A. So so we need to multiply these number by 2, and if we want the per glucose ale. So for one cycle it is 12 as we have discussed, and if we multiply it by 2 then we will get 24, right? So now your brain might stuck question, where is all the ATP, right? You may thinking that the ATP output of the citric acid cycle seems pretty unimpressive, right? All that works for one ATP or GTP. Okay, that's true that citric acid cycle does not produce much ATP directly. However, it can make a lot of ATP indirectly by ways of NADH and FADH2 it generates, right? These electron carriers will connect with the last portion of cellular respiration, depositing the electrons into the electron transport chain to drive up synthesis of ATP molecules through. Okay, so I hope you enjoyed the session, right? So we have 8 main steps of this cycle, citric acid cycle, right? And 24 ATP, right? And the citric acid cycle mainly occurs in the mitochondria, matrix of mitochondria, right? So one last point, in eukaryotes, the citric acid cycle takes place in mitochondrial matrix, just like conversion of pyruvate into acetyl coenzyme A, right? But in the prokaryotes, these steps both take place in cytoplasm, right? So yeah, this was from my end of citric acid cycle. If you want to learn more about citric acid cycle or the biochemistry, please book a session on Learn Classic Education. Okay, For now, bye bye. Take care.